Welcome to the third part of the module 4, where we are going through the quality assessment of raw sequences. There is a saying that garbage in, garbage out. In the same line, it takes a good lab practice to produce lab data for the downstream processing. If we mess up in the wet lab, it cannot be corrected in any way in the dry lab. This concept is very critical, especially to all researchers to understand, because if for some reason there was some problem that was introduced in the dataset during the lab processing, like the PCR or the labeling, for sure this mistake will be carried on to the sequencing platform and later to the bioinformatics, and it may end up with the wrong result from a given research question. So it's very important that we check the quality of the raw reads before we do further processing to ensure that we don't waste energy and resources to process the data that will make no sense. So when you get raw sequence ready from your client, normally they are compressed, and you will get, for instance, in this case, you have a paired end reads. You'll be getting the forward read, here named R1, and the reverse read labeled as R2 for the same samples. And also for the second sample, you have the forward and reverse read. But if you open any one of these samples, you'll be able to see that we get a sequence, we get an information where the first line starts with a letter 80, with the H character, and is followed by a sequence identifier and an optional description like a faster title line. So the first line will be giving starting with the H and a sample identifier, and the second line is a raw sequence letters. These are the different nucleotides that are present in that given sample. So this is the sequence nucleotide, and the third line beginning with a plus character and is optionally followed by the same sequence identifier like in the first line. And the fourth line, most importantly, is the quality score of each of those nucleotides in the second line. And it must contain the same number of symbols as the letters in the sequence, because each nucleotide in the line number 2 is having its own quality score, which should be reflected in the fourth line. So to judge the quality of each sequence, researchers have come up with the so-called Phred score. The Phred score describes the quality of the sequence and is presented by an integer value. The larger the value, the more confident there can be in the output or in the sequence generated. And the probability that the base is wrongly called or incorrectly called is given by the formula P is equal to 10 power minus Q over 10. If you transform this by introducing logarithm, you get that the quality score Q is equal to minus 10 log P base 10. For example, if the probability of an error P is equal to 0.01, which would mean that the chance that the base is correctly called is 99.99, and the chance that is wrongly called is 0.01, then we'll be able to get the quality score of 20. But what if the P is equal to 0.0004? What will be the quality score? Of course, if you plug in there, you'll find that the quality score will be 40, meaning that there will be 0.0001 chance of a base in particular sequence to be wrongly called. And these symbols that reflect the quality score, normally they are generated from ICC number. These are American Society of Communication for Information Interchange. As you can see here, for instance, the 33 here presented by exclamation mark means that the probability that the base is wrongly called is 1. And then if you put there in the question, you get that the quality score will be 0. Likewise, if the chance that the base is wrongly called is 0.1, would mean that the quality score will be 10, and in the fourth line is presented by a letter plus if you revisit those characters in the third and the fourth line. So it's highly recommended to check the quality of raw sequence read before further processing of the raw sequence read. This will save much time to be invested and the resources to work on the data which actually would make no sense after the bioinformatics processing. So there is a tool that works very fine to assess the quality of raw sequence read. This was developed by Andrew in 2010. It's a very powerful tool and it's open source. It works very fine to assess the quality of high throughput sequence data. You can find this tool in the link provided here. And from the same link, you'll be able to see example of the good Illumina data as well as example of the bad Illumina data. And I've generated some of the sample output from the FastQ plot. For instance, this is the one you get after getting the output. To give you, you get the samples, the forward and the reverse read. In case here we have the second and the forward and the reverse read, but then it gives you the best statistics about the sample, where for instance you can see here the file name or the sample name, you can see there, and the file type is conventional base cause. The encoding in this case was Illumina, and the total sequences per that zip is 19,096, almost 200,000. And the sequences flagged as poor quality is 0. The sequence length is 251 and the GC content is 51. This gives you an overview of the quality of the sequence that you have processed. And if you know for sure what would be the sequence length, then you would judge by seeing what you get from the FastQ output. I've collected some of the sample output, for instance, for the forward read. One of the sample, the forward read, you see, normally when we get the raw read, we expect the quality of sequence to be further processed should be at least with the quality score more than 20. So you see in this forward read, you see the quality score was above 20, except toward the end, the quality was dropping below 20. Likewise, when you see the reverse read is too much of poor quality toward the end, and this is very common because for the high throughput sequence data, the reverse read generated by the reverse primer normally brings poor quality toward the end because of the fact that the reverse primer is having the barcode or the index that would be used to multiplex the sample. So this is expected to get a poor quality toward the end of the reverse read as opposed to the forward read. What would you say if you find maybe this is your raw sequence data? You see here the sequence was 301 lengths, but you see the quality toward the from the beginning was of good quality, but around 50% of it, the quality is below 20. So this quality is very bad. You cannot get anything out of this even if you process with the high throughput tools. For that matter, you must figure out if really it's worth going on working with that kind of data. If I were me, I would straight away reject this kind of sample because the quality is very poor and I don't expect to get anything good out of it. But what about this kind of output? This is very good quality. They are all above 40 quality score. This actually was generated after merging the forward and reverse read and trimming all the poor segment from both ends, beginning and toward the end. Normally at the beginning of the sequence will get poor quality because the reagent when you start the amplification are not yet optimized, so the quality will be a little bit lower. But toward the end, when the reagent are exhausted, now again the quality of the reads are not that much good. So when you trim those ends, then you match the forward and reverse read, you get a very good quality like this one, which actually assures you that you are in the right track and you are likely to get good output out of the data. So let me end there. We'll proceed now to see the bioinformatics part in next session. And at this moment, let's end this part. So see you then.